Okay, so for our next session, by the way, there's still people coming in. Okay.
Yeah, I'll let them in as they come, so you can continue.
Shall I meet them as they come, or you are going to do that?
No, I'll do that, doctor. You can,
okay,
yeah, I'll do that.
Carry
You
on.
can continue the lecture.
All right, guys.
Yes.
Now we'll be talking about three very important cells in the male reproductive system. These cells are Leydig cells, Sertoli cells, and spermatogonia. The portion of the testes that produce sperm and secrete testosterone are structurally and functionally distinct. And wait a minute, let me minimize this window. Go away. Okay. And okay, where were we? Okay. Now, 80% of the testicular mass consists of seminiferous tubules, within which spermatogenesis takes place, supported by Sertoli cells. Now, 20% of the connective tissue is where testosterone is being produced and is where Leydig cells reside. The Leydig cells, or in another name, interstitial cells, are part of the endocrine cells that produce testosterone and lie in the connective tissue between seminiferous tubules. Under the influence of the luteinizing hormone secreted by the anterior pituary, as well as the adrenocorticotropic hormone, cholesterol is being transformed into its active form, testosterone, and in specific tissues where 5A reductase amino acid is present, we have the conversion of testosterone to dihydrotestosterone. Now, you have to understand that testosterone and dihydrotestosterone are slightly different, and they cause different changes when it comes to the male body. Although testosterone may be particularly important at the early stage of life, dihydrotestosterone is what is going to lead to the maturity of the male reproductive system. So over here we have what we spoke about more or less before about the positive feedback and the negative feedback of the hypothalamus and the cells that exist in the seminiferous tubules or the testes, let's say. So under the influence of hypothalamus, there is the release of gonadotropin releasing hormone, GnRH. Now, GnRH is positively gonna impact the anterior pituitary for the release of two hormones, LH for the Leydig cells and FSH for the Sertoli cells. Now, LH is gonna prompt the Leydig cells to compose testosterone following by breaking down cholesterol and transforming it into its active form. Now, testosterone, and in the occasion that I do have testosterone receptors in the anterior pituitary and the hypothalamus, they're negatively gonna affect the production of testosterone. They're gonna indirectly affect the release of LH as well as FSH by suppressing GnRH at the hypothalamus, and they will directly affect LH levels by inhibiting the release of LH by the anterior pituitary. Now, in addition to the Leydig cells, we have the release of inhibin by the Sertoli cells. Now, inhibin is mainly, its action relates specifically to the anterior pituitary, and what happens is that it will inhibit the release of FSH. Sertoli cells compared to Leydig cells are more of a support cells. They're not so endocrine cells. they mainly support the function of spermatogenesis. Do you have any questions in regards to the negative feedback of the male reproductive system? Okay, it's quite easy.
It doesn't look like there are any questions.
Okay, fantastic. Okay, it's quite easy. You can also read it on your own. They're just direct and indirect effects that you need to know, and specific hormones that are being released. Now, spermatogenesis takes place inside the seminiferous tubules, in which we have spermatogonia that provide for us a continuous germ line for the differentiation and packaging of the spermatozoa that will later on be part of the semen. Each spermatogonium has a diploid strand of 46 chromosomes. By the end of the mitosis and meiosis, each spermatozoa has a haploid set of 23 chromosomes. So we go through mitotic proliferation, and each time there is a mitosis going on, a primary spermatogonium is gonna give me another primordial cell. However, in addition to this, I will also have another daughter cell identical to the first one sitting on the outer edge of the seminiferous tubules. In this way, the germ cell line is always maintained and the genetic material is never altered. After the mitotic division is completed, I have the meiosis that is basically gonna happen. So during the first meiosis, I will have a primary spermatocyte that is going to have 46 chromosomes. That is going to have a haploid double strand of 23 chromosomes. By the end, it's going to give me, in its second meiotic division, it's going to give me 4 spermatids of a single strand of 23 chromosomes. These eventually, one of these spermatozoa in the millions that will be produced by this germ cell line is gonna fuse with the nucleus of the ogonium or the ovum to give me the zygote. Production of spermatozoa or of spermatids, it takes some time. It doesn't only, let's say, go from mitosis to meiosis, but it also needs a lot of remodeling or structure. This happens through the support of the Sertoli cells, but also chromosomes, let's say, or sperms that have the X chromosome, they stay attached to also other cells. So all these spermatids that have been produced by the meiosis or mitosis, one of them is definitely going to have the X chromosome. Now, all of them will be connected between them through a cytoplasmic bridge. And they do that because they want to maintain the exchange of specific genetic material, because only the X chromosome and not the Y contains information in regards to testosterone. And because testosterone is super important for the remodeling of the spermatid interspermatozoon, it's important that all spermatids, they stay connected between them throughout the remodeling phase. Okay, so the spermatozoon is made up of 4 main elements. The tail, which is basically gonna give the propulsion and it's gonna stay static for some seconds after being deposited in the cervical canal. We have the acrosome, where inside it we have the acrosomal enzymes that they will provide the drill or the force in which the ovum is going to be penetrated. And finally, we have the midpiece, which is basically the energy center of the spermatozoon that is going to give it the energy to be able to travel to the ovum. As the sperm is being deposited in the female reproductive tract, it goes into a period of capacitation, and this may last for 4 to 6 hours. Capacitation is a period which is characterized by the retraction of cholesterol and seminal fluid is washed free. And in addition to this, surface proteins are being redistributed. So the acrosomal membrane fuses with the outer sperm membrane, eventually creating a poroid plexus of the head that is from where the drilling enzymes of the acrosome are going to escape to penetrate the ovum in occasion of fertilization. Now, all these sperms that are dormant currently during this phase, we start their whip-like fashion of swimming towards the ovum after a calcium influx that is being released by the mature ovum. So through this calcium channel, through these calcium receptors that exist in the spermatozoon, the tail is activated and the big swim starts towards the ovum. Okay, let me check your questions really quickly.
Yeah, Doctor, one of the students was asking a question. Do you mind if I read it out?
Yes, sure.
All right, so she was asking, do the gonadotrophs of the anterior pituitary secrete both FSH and LH at the same time?
Yes, so the anterior pituitary secretes LH and FSH at the same time, yes. So if you see testosterone mainly, okay, so sperm matures every 64 days, let's say, we have a new line of sperms. However, testosterone produced by Leydig cells has a negative inhibition directly on the anterior pituitary, so it will suppress FSH and LH. So without testosterone, let's say sperm cannot mature. It needs to bathe in testosterone in order to be remodeled. So the question basically was if FSH and LH by the anterior pituitary secreted in the same amounts, right?
She was asking at the same time.
At the same time.
Yes, doctor. She was asking if there was
it's secreted at the same time.
Maybe she's referring to this.
Yeah, it's a simple answer. It's secreted at the same time.
All right, perfect. Thank you.
Okay, we're good to go? Have the green light?
Yes, we're good to go.
Thank you.
Yes, all good.
All right, close this. Okay, now over here we have a diagram of the seminiferous tubules. Over here we have the sustentacular cells, which are basically Sertoli cells that support the production of sperm. And we also have the interstitial cells or Leydig cells that are my endocrine cells that are gonna give me the testosterone. And through the exchange of information with the Sertoli cells, I have the production of sperm. Now, the cytoplasm, as you can see over here, of Sertoli cells engulfs or envelops the production of sperm. So as all this go through a process of remodeling, so these are dividing spermatids over here, they reside during this remodeling with the Sertoli cells. The reason is during this process of spermiation and the release of the sperm in the lumen and the production of aqueous fluid by succinticulate cells, which is going to push the sperm in the epididymis and from the epididymis to the vas deferens, sperm is being fed by Sertoli cells. Because sperm cannot take up glucose, Sertoli cells play the role of the mother. Thus, they take glucose and they turn it into lactate, eventually nourishing the sperm. In addition to this, through the process of remodeling, there is a lot of cytoplasm that is being extruded by the spermatids. So, in addition to this, Sertoli cells have a phagocytic function. Very importantly, Sertoli cells create the blood-testis barrier that prevents any forms of substances in passing and altering the genetic material. And this barrier is being present in two places in the human body, in the cortex and in the testis, or let's say the ovaries. The reason is because there are two very important functions for humans: procreation and also the central nervous system, which guides the rest of the body. So these two need always to be well guarded. As we said, Sertoli cells secrete the seminiferous, the seminiferous fluid or the aqueous fluid that is going to push the spermatids to the urinary tract. And they also produce the androgen binding protein, which along with the testosterone production by Leydig cells is gonna bind testosterone and is gonna nurture the sperm for the remodeling process. So testosterone is very important for the remodeling. In addition to this, Sertoli cells, as we said, they're gonna produce inhibin, which is gonna directly suppress the FSH levels. It's not going to act directly on the anterior pituary. However, it's mainly going to affect FSH level production. So this brings us to the end of our session. We have another lecture coming up right now, which deals with ejaculation and
Hi, Doctor. Doctor, before we move on, there's one question. Should I read it out?
Yes, please.
Yeah, so one of the students asking, what is the significance of androgen binding protein?
Now, the significance of androgen binding protein being secreted by Sertoli cells is significant because only under testosterone sperms mature. So sperms will start differentiating and maturing and being remodeled only under the influence of testosterone. So they are able to coat testosterone, and they know that only under an environment of testosterone can they differentiate, get remodeled, and get ready to become mature in order for them to be able to procreate.
Hi, Doctor. And there are two more questions. One is one of the students saying on slide 16, it mentions that chromosome X, not Y, contains genetic info crucial for sperm development. So he's wondering, isn't the Y chromosome the important one, or are both of them equally important?
No, it's the Y chromosome. There's a mistake on the slide.
All right, perfect. Thank you. And the last one, from where the calcium secreted incapacitation?
Sorry? Okay, calcium.
Could you please, yeah.
Calcium is secreted basically by the ovum. So even if you put sperms in a slide and you just put some calcium, whatever, there is in the plasma in which the semen is swimming, you will see semen swimming, all of them in one direction. Okie doke. Next one.
All right, perfect. Thank you. Yes.
Okay, sexual arousal and ejaculation. So when it comes to sexual arousal and ejaculation, there are certain elements that are very important. And these elements, they mainly relate to the semen, where it's being deposited, the accessory sex glands, where it's going to give us the bulk of the semen, and also the engorgement of the male penis that is going to provide the vessel through which semen is going to be deposited in the cervical canal. There are different phases of the sexual act, or let's say the engorgement of the penis, which includes ejaculation, emission, and expulsion, and also the refractory period. So let's look into our lecture. Let's have a look into our lecture now. So the male reproductive system is made out of a complex pathway of tubes, as you see from the epididymis, vas deferens, all the way to the urinary tract. Over here, we have the seminal vesicles that they're gonna give me 60% of the seminal volume. I have the prostate gland, which is a very important gland, and you may have heard in clinical scenarios. And also we have the bulbourethral glands over here, or Cooper's gland, which is basically gonna give the lubricant for the culminating act. The penis is a muscle modified to enter the female reproductive system, and it's shaped in such a way to provide not only traction but also provide some form of seal between the cervix, the vagina, and the male penis. Now, the epididymis, it's a structure that is loosely attached on the testes, and it's something that is it can be felt if palpated. And next to the epididymis, there is a muscular tubule called the vas deferens. Now, the vas deferens is an important structure for the reason being that in that specific structure, where it exits the scrotal sac through the inguinal canal and deposits the semen in the head of in the edge of the bladder, or the urine, or on the neck of the bladder, it also has some additional functions. It retains or it bathes the semen in testosterone before it exits the male reproductive system. In addition to this, it provides a form of defense mechanism through a specific enzyme called defensive. Thus, not only it protects the sperm, but also it gives the sperm a motile element. So it's basically, as after it's being deposited, this is also one of the factors that need to be in place for the sperm to be motile. And as it's being deposited in the vas deferens, is being nurtured by simple sugars that exists in this specific vesicle, and they may not be a lot, let's say, over there at the specific site, but because the semen is non-motile and is also very densely packed together, it does not need a lot of energy in order for it to be retained. in the occasion that men decide to let's say stop producing, stop having the ability to procreate, there is a procedure called vasectomy, in which the vas deferens that is going to pass from the inguinal canal to the neck of the bladder is going to be disrupted surgically. Now, the buildup of sperm in the vas deferens, is removed by the process of phagocytosis. Because I had this question in the past, there is also the potential to reverse this procedure again through surgery. So this could be also reattached, let's say. They could again be reattached. So there's three glands when it comes to the male reproductive system. One of the, not the most important one, the one that provides the biggest bulk of the semen are the seminal vesicles. The seminal vesicles that are very close to the ejaculatory duct, during the ejaculatory phase or the excitement phase, they start producing the fluid that is necessary for the sperm to be diluted and be deposited in the female reproductive system. In addition to this, they provide fibrinogen, which is going to create the meshwork that, along with the secretions of the prostate, they're going to clot the semen inside the cervical canal to be able to be retained after the retraction of the penis from the vagina. So these two, along with the prostate, they play a very important role. Now, liquefaction elements come later on, as we're gonna see, from the prostate, to relate back to your PBL case. In addition to this, the seminal vesicles produce prostaglandins. That prostaglandins, if you attach them or if you apply them in exposed muscular tissue, it's going to lead to contractions. This is also particularly useful for the uterus because it's going to help the sperm reach the female oviduct for fertilization. Now, over here, we see the prostate gland, which is basically surrounding the neck of the urinary bladder. In the occasion that the prostate goes through a process of hypertrophy, particularly this may happen in an older age, then the urinary canal is it becomes particularly narrow, eventually leading to painful cessation during urination. This can be reversed by applying or by consuming or taking, let's say, prostate-specific antigens, which is gonna lead to the dissipation of the swelling of the prostate gland. So the prostate will produce the alkaline fluid, which is gonna break up the acidic environment of the cervix. So this fluid is particularly alkaline. It provides clotting enzymes, which, along with the fibrinogen, is gonna give me the seminal clot. And in addition to this, is gonna provide prostate-specific antigens, which are gonna break up the seminal clot for the sperm to be released and enabling it to swim all the way to the ovum. The Bulbourethral gland, it's a gland that plays a role mainly at the beginning of the sexual act, and it provides the lubrication to avoid friction between the vagina and the penis. However, the secretion, let's say, the bulk of the semen is not so dependent on the secretions by the Bulbourethral gland. So now we finished in regards to the composition of the semen. We're gonna look into the elements that lead to the deposition of semen into the cervical canal. In order for this to happen, erection needs to take place. Erection can be facilitated either psychologically or mechanically, because the erectile tissue of the penis, it's dense when it comes to mechanoreceptors. Thus, a small activation or small stimulation will lead to the erection of the penis. The second act of that is being facilitated by the external male genitalia is ejaculation, and basically the expulsion of the semen from the ejaculatory duct to the outside environment. So there are 4 phases that they're taking place during the sexual response cycle. There's the excitement phase, which includes the erection and heightened sexual awareness. The plateau phase, which is the hardening of the male external genitalia, and there is also an increase in heart rate, blood pressure, and respiratory rate. The orgasmic phase, which includes the ejaculation of which includes the expulsion, or let's say the release of the semen, along with a certain culminating or mounting sexual excitement, in which involves the rhythmic throbbing of the pelvic region, both in male or female, and the resolution phase, which involves the restoration of the male genitalia to their original form. So when it comes to the penis is made up of 3 sponge-like vascular spaces. During the excitement phase, which is governed by parasympathetic nervous system activation, we have vascular dilation of the arterioles that you see, we're gonna see over here later on, facilitated by the local release of nitric oxide. And as nitric oxide is being released, then the arterioles expand, and they're able to deposit much greater amounts of blood in these spongiform spaces. Now, as this spongiform space expands, then the arteries or the venules that will drain blood from these spongiform spaces go through a process of compression, leading to what we call penile vasocongestion. So the vascular spaces are so filled up now with blood that that arteries are being compressed, eventually leading to an increase of arterial pressure in these vascular spaces, engorging the penile tissue, altering its color, and also inhibiting venous return because of the compression of the veins. Now, we spoke about this. Now, in the occasion that we do have an erectile dysfunction or impotence, as it's being called, this could be driven by 2 different factors. It could be driven by either physiological reasons, such as, let's say, problems with the arteries, or it could be the individual could be going through chemotherapy. Other factors such as diabetes, which leads to problems with the arteries. However, it could also again be facilitated by psychological factors. Now, it's kind of a chicken and egg question, because having erectile dysfunction would lead to anxiety and stress, and also vice versa, having anxiety could lead to erection and erectile dysfunction. Erectile dysfunction is much more prevalent in older ages. And the reason is because that usually when when the penis is engorged with blood, in order for this, in order for the blood to be dissipated and be redistributed, then we need the action of a specific enzyme. And this is the cyclic GMP. Now, if the cyclic GMP is not present, or is being suppressed and not broken down, then it will lead to greater amount or greater time of erection. So let's say we have the release of nitric oxide, arterial dilation, the deposition of blood into the spongiform spaces of the penis, and then we have the activation of the enzyme cGMP or cyclic GMP that eventually is going to break down the erection. Excuse me, sorry. We have the enzyme PDE5 that is going to break down the erection. Now, there are certain drugs such as sildenafil that will inhibit PDE5. So by inhibiting this enzyme, then erection can be sustained for much longer. So during the ejaculatory reflex, we have the delivery of fluids in the ejaculatory duct, starting from the prostatic fluid, then sperm, and finally the seminal vesicle fluid. All this activity, however, before the deposition of, before the ejaculatory reflex or the emission, if you may say, is being preceded by the secretion of lubrication by the Bulbourethral gland or the Cowper's gland. So expulsion after the urethra fills in with semen, the sperm is forcibly expelled through the rhythmic contraction of the pelvic region. During this physical activity, there is an involuntary throbbing of the pelvic muscles, generating certain muscle contractions either in the penis or along with the pelvic region. And after this phase passes, we have the resolution phase, in which we're going to have the activation of specific enzymes that are going to break up the arterial dilation and eventually restore the penis back to its original phase. It is very important that the semen has a large concentration of sperm in its count, for the reason being that there need to be a lot of acrosomal enzymes to break down the ovary's defenses. Thus, it is not only just one semen that is going to go and attach itself to the ovum. It's kind of a group activity. So now we're ready to answer any questions that you guys may have. I see 6 in the chat.
Does anyone have any questions? If they have, they can write in the chat. For Abdullah, you can clarify a question. That would be great. Anemia would cause what?
Erectile dysfunction.
Okay, then yeah, probably that's what he's asking. Yeah.
Okay, so what I'll do, I'm just gonna correct the XY chromosome that we spoke before and re-upload again your lecture slides. If you have any question, please email me. I will try and get back to you during the weekend. And hopefully we might have a review session to solve any questions that you may have in regards to today's lectures. I hope you enjoyed it.
Doctor. Yeah, it's just Abdullah's question. Yeah. Doctor, we have just one question. Students asking, would anemia cause erectile dysfunction?
Excuse me?
He was asking, would anemia cause erectile dysfunction?
Yes, it would. I mean, it depends. If not necessarily, let's say, but it's gonna affect the blood dynamics as it does in the rest of the body. So because it does the same in the rest of the body, it's potentially gonna do the same in the erectile tissue. But please take my response with a spoon of salt, because I'm not an expert when it comes to this. So maybe a blood specialist would be able to answer this question much better than I do.
All right. Thank you so much, Dr. Stephanos.
Thank you, guys.